These are notes on DNA and gel electrophoresis to help you understand the DNA lab that you're going to do later this unit. First, I want to do a little bit of review on what DNA, or genetic material, as Amplify often calls it, actually is. So you've run into DNA, again, or genetic material several times this year. And I wanted to just remind you of where that was. So the first time was in the light waves unit here, where we learned that that damage to genetic material, or DNA, is what can cause cancer. And then in the traits and reproduction unit, we saw that in the nucleus are these things called chromosomes that had genes on them. These little yellow bars are supposed to be where the genes are. And that those genes were the instructions to make proteins that then give us our traits. Well, what genes actually are segments of DNA, or segments of genetic material. And one long strand of DNA is called a chromosome. And that chromosome contains many genes. And by many, I mean, depending on how long it is, it could be dozens, but it could also be hundreds. And so this purple thing here, that is one long strand of DNA, and that is called a chromosome. So you've run into DNA several times this year, and now we're going to see how the amount of DNA two organisms share can tell us how related they are to one another. And this makes sense because we've already talked about how if two organisms share a common structure, so say 4 limbs or fur or something like that, those are traits. And so they must share DNA that leads to those traits. So that all makes sense, except that by using DNA, we can get to an even smaller and smaller gradation of how closely related they are. So we could take all these things that have fur and then analyze their DNA and see which ones are most closely related, for example. So what we see is we can use DNA to discover relationships. And what we know is that proteins are made up of smaller parts called amino acids. We learned that during the traits and reproduction unit. And the more similar the amino acid sequence, the more closely related the two species. So remember that genes are the instructions to make proteins. Proteins are made up of amino acids. So really what genes are the things that tell the amino acids in what order to form or to organize themselves. And then from there, the protein makes its little shape. But that means that if amino acid sequences are similar, then the DNA has got to be similar. So down here, you don't need to worry what these mean, like the lys, the glue, the his, the iso. They're just abbreviations for different amino acids. It really doesn't matter what those amino acids mean right now. But these are all looking at the same part of one gene. So they're all the same gene, but remember, we can have different versions of a gene. So they can be a little different from one another. And this happens to have to do with the hemoglobin molecule, which is the molecule in red blood cells that carries iron. So if we look at like human here and chimpanzee here, we notice that they're actually identical, right? They both go lys, glue, his, iso, lys, glue, his, iso. So over here, it says the number of differences, there's 0 differences between them. But then if we look at human and gorilla, or we can look at gorilla and chimpanzee because they're exactly the same, we notice they both go lys, glue, his. But then the chimpanzee and human are iso and the gorilla is lys. Okay, so we say there's 1 difference, right? And if we keep going, right, chimpanzee and horse have 3 differences, horse and gorilla have 2. So we can look and compare these differences and say, so the human and chimpanzee have to be most closely related. The horse and chimpanzee, or horse and human, have to be least closely related, and everything else falls in between. So if we were to go and make a phylogenetic tree based on that DNA, it might look something like this. So remember, these are the number of differences here. And then I've put in some pictures, right? All of these are related. They're all mammals. And so they are all related in some way. Realistically, all life is related, but right, these are more closely related than they are to, say, worms or insects or trees or bacteria, because they are all mammals, they're all vertebrates, they're all quadrupeds, meaning they all have 4 limbs. So lots of relation here. But we can see here we have a human. This is my older daughter, Mia, in a Harry Potter costume when she was little, and a chimpanzee. And we know these two are most closely related of all of these. Okay, so that's why, remember, this is going to be, if we're looking at a time scale, this is older and this is current. And so a long, long time ago, these were all the same organism before they all branched off. And then we have the horse and zebra kind of going off on their branch, and then the gorilla and chimpanzee and human going off on their other branch. And this is the primate branch here. Okay, this is the equine branch here. You don't need to remember that, but that's how those two split, right? Then we have the horse and the zebra splitting off. But notice I specifically put that at about the same branch level. I was drawing this, so it's not perfect, as the gorilla splitting off from the chimpanzee and human, because over here, human and gorilla are separated by one difference in that set of amino acids, as are the horse and zebra. So they're, kind of related to the same degree. Whereas the human and chimpanzee are a lot more closely related, meaning they split off from each other more recently and sharing more recent common ancestor than they do with the gorilla, or than the horse and the zebra share. And so you could build a phylogenetic tree that way based on looking at the DNA. So, kind of take a look at that, make sure it makes sense to you how I did that. All right, so that brings us to gel electrophoresis. So gel electrophoresis is a process by which, instead of having to analyze each amino acid sequence, which is very complex, very costly, you can actually analyze chunks of DNA to see relatedness. We'll just say that for right now. So over here, this first picture here is a gel electrophoresis machine. And this is a power pack here. So electrophoresis has to do with running an electric current through a gel box. This is the gel box. That gel box is filled with a chemical buffer, so a liquid. And that chemical buffer keeps the pH, the acid-base amount, relatively stable. Otherwise, normally when you run electric currents through things, you get a big difference in acid and base concentrations. So the buffer is going to keep it relatively stable. Again, this is more detail than you need, but I know some of you get really curious about this stuff. And then in here, what you can't see right now is what we call a gel. And this gel is going to feel a lot like pretty thick Jell-O, okay? Here we can see it a little up close. Again, it's a little hard to see, but you can see that somebody is using a micropipette. We used these during traits and reproduction when we genetically engineered our bacteria. And this person is putting little samples of DNA into little wells. So these wells kind of look like this, okay? And then you take your micropipette, have a pen, whatever, and you kind of put it in the well. You don't want to stab it through the well or all of your substance is going to fall out the bottom, but you kind of put it here and you press it to let it go, and it fills up the well with, these nice little kind of blue, like it'll look blue. DNA itself is actually colorless, but we're going to use a blue dye so that you can see it, because asking people to put a colorless substance into a gel that is also vaguely colorless, who would know? You know, half the time we'd be like, I don't know if it's there. So at least if we see this, we know, okay, we got the DNA where it's supposed to go. And we call these wells. So in this gel, let's see, they're filling up right now, well number 7. Okay, we'll have 8 wells because we have 8 different organisms. And then here, this is what the gel is going to look like once it's done. And it is going to kind of glow like this. As I said, DNA is colorless. We're going to be using a radioactive tracer that shows up under ultraviolet light. And so it is going to kind of glow like this. Ours looks more orangey, but that's going to be kind of what it looks like. And so now I'm going to go into more detail about how we do all this. So in each human cell, there's approximately 6 feet of DNA in each cell. So I like this analogy of a cassette tape. So this is prehistoric music, like, you know, from when I was a kid. This is all the tape. It was called tape. Music was coded on this magnetically, actually. But this is all the tape that fits neatly into this little cassette here. So I like to think of that as all the DNA in your cells. And your cells have really creative ways, kind of like these little spindles here. They would turn and you could roll up the tape every now and then. Your tape player would eat your tape and it would come out. I mean, this is kind of a disaster, but a bunch would come out and you'd have to sit there with a little pen and kind of twirl it back up. But think of that as all the DNA in your cells, okay? So if you just were to take that DNA and you put it in the little wells, you get these huge, weird kind of smears of DNA, and it doesn't really tell you anything. So what we need to do is we need to chop the DNA from each organism up into pieces. So what we do, and we're going to do this before you even get to it, but we're going to mix the DNA with some enzymes. And what enzymes are proteins. Here again, we see some proteins that cut the DNA at certain bases to make DNA fragments. We have DNA from 3 different organisms. So we've got organism 1, organism 2, and organism 3. And let's say we're using an enzyme that cuts DNA wherever you see CCGG, and it cuts it between the C's and the G's. So if I'm looking at 1 here, I would put a cut here. My enzyme's going to cut there. Then I'm going to keep going, and I see I'm going to have a cut here, and I'm going to have a cut here. So in this length of DNA, I would actually end up with 4 little fragments of DNA. This one would be the smallest here, and this one would be the largest here, and the others would be in between. So if I've gone through and cut all my DNA, what I can see is that organism 1 and organism 3 have fragments that are very, very similar in length to each other, which means they are more closely related to each other than they are to organism 2. So I might make a phylogenetic tree that looks kind of like this. And that's how I would use the DNA. So what the gel electrophoresis is going to do is it's going to divide the fragments up based on fragment size. And I'll show you how that's going to work. I'm going to show you now a little more detail in how to run and then read the gel. The filled gel is in here, and this one looks like it hasn't been filled yet because you can't see the nice little blue wells filled, but that's okay. And those wells are actually going to be filled. I'm going to make them red right now just because I happen to have the red here, or maybe not. Let's see. Oh yeah, there we go. Those get filled on the negative side of the gel box. So the black lead here, there's a black wire and a red wire. The black lead goes into the negative electrode, and the red goes into the positive electrode. DNA itself is negative, so negative attracts to positive. So the DNA is then going to run in this direction as we put the electric current through the DNA. And it's going to run, obviously, all the DNA runs for the same amount of time. And so what we have here would be 4 different samples. And so this would be like organism 1, organism 2, organism 3. I can put that in here. So that's 1, 2, 3, and then 4. And then each sample has been digested by enzymes, so has the different fragments in there. Okay, so when you read this later, this gel here, this is before we used the radioactive tracer. Don't worry about what the A means. That was from a totally different project that we used to do years ago. Ours will look like this, but it'll be glowy. This was when we used more of a solid dye. Doesn't matter. But when you look at this, you see different bands. And when you read it, you're going to read it going down. So here's the well here where the DNA was. These are all the fragments that were in that well from that organism. So if I look back here, right, organism 1 here had 4 fragments in it. Organism 2 had 3 fragments in it. So if I go back, you know, and these are not the same thing, but this might be what organism 1 looks like, because there's 4 fragments. We say bands. So here's 1, here's 2, here's 3, here's 4. And those bands are organized largest to smallest. So if you think about all of these fragments physically going through this gel, I like to think of it as people holding hands running through a forest. Those groups that are largest, say 8 or 9 people holding hands, are not going to get as far as those groups that are smallest, say 2 people holding hands, right? 2 people holding hands can get a lot further through a forest running than 8 or 9 people. They're just going to go more slowly. There's, you know, more things to bump into. So it's going to be harder for them to move forward. So the larger fragments end up staying closer to the wells, and the smaller fragments end up migrating further from the wells, which then means that lanes, so these are the lanes here, the like 1, 2, 3, the lanes where the fragments are approximately equal are going to be more closely related. So if we look at lane 2 here, and then we look at lane 4 here, we see that those bands, like here and here, here and here, here and here, and then there and there, are very similar. And so we know 2 and 4 must be pretty closely related. And so that's how we can use the gel, which is based on the DNA, to tell how closely related organisms are. So that is a huge overview in a reminder of what DNA is, why we can use it to tell relatedness, and then how we can use it to tell relatedness. Please make sure you are asking questions in class so that you really understand how this all works. Have a good day.